Okay. It's just a short introduction. So today I'm very excited to have Ethan Ancell from Daniela Witten's lab presenting today. He's been working with a few groups here at NAPE, and in particular, today he'll be talking about collaboration with Madeline and Madeline, the graduate student in Garrett Stuber's lab. And the talk title, or the topic that Ethan will be talking about is applying generalized linear models to data that involve behavioral and neural activity recording components. So with that, Ethan, you can take it away.
Yeah, sounds good. I'll just get my slides up and hopefully everything looks fine. Can you see my?
Yeah, that looks good.
Okay, and it's not the presenter view. It's like
yeah, it's not the presenter view.
Okay, good. Yeah, so thanks for the introduction, Charles. So I'll be presenting on this talk, determining statistical significance in behavioral neural activity linear models. This is joint work with Daniela Witten in the Department of Statistics and Biostatistics, and also a collaboration with some NAPE people, which I'll talk about. Also, I personally am very okay if people step in to ask questions during the talk. Pretty low key and fine. I probably won't see it if you type it in the chat just because the view of Zoom's not great, but feel free to speak up and that's totally fine with me. So
I can monitor it and let you
oh
know.
yeah, or you can monitor and let me know.
Yeah.
That sounds good too. Yeah, so just a bit about me. I'm a first-year graduate student in the Department of Statistics in a PhD program. And the funding for my graduate studies coming from NAPE. So you guys are paying for my degree, so thank you. And I'm excited about studying statistics with applications to neuroscience. And I'm interested in lots of these types of problems and open to collaboration. So if you have an experiment that you're running or et cetera, and you're interested with statistical or interested in the collaboration from a statistical point of view, I am open to those. And you can contact me at this email, ancell@uw.edu. And so this particular talk was some work that was done with Garrett Stuber and Madeline Hjort in the Stuber lab. And their research was in understanding reversal learning using 2-photon calcium imaging technology. And my collaborators are awesome. They're also I guess they're your colleagues as well. So they're great, and big thanks to them. So next I'll talk about the technical details of this talk. So just the setup, groundwork of what we're talking about. So this is a stats talk, so we're gonna have a stats model here. And we're gonna write is this y1 to yt, and we're gonna write this little linear model thing, but I wrote it in matrix form. And so to clarify what each of these little pieces are, because we're neuroscientists, in this particular case, we're gonna be looking at DF over F recordings, and we'll just consider this to be for a single neuron. And we're gonna say that there are capital T time points recorded in this particular experiment that you're running. And in addition to this, we also have this big matrix of covariates or predictor variables, whatever you want to call them. And there's capital T of them, there's little p of these predictor variables, but we have essentially capital T recordings. So we have one row for every single time point in our experiment. So the sort of situation we'll be operating under. And we also have this little vector of coefficients to make this a linear model. And then we just have this little vector of T error terms. So this is essentially the part of Y which is not accounted for by this linear model of X, or linear model in X. So in this particular setup with Madeline and Garrett's data, they had a special structure on X. This wasn't any old X. They actually had it chunked up in a particular way. So they had this little, they took X and essentially a few of them were designated to different tasks. So maybe this X1 was particularly odor chunks or an odor chunk. And the next one was a licking chunk and other variables they want to record. And then just essentially we're leaving this a bit abstract. We're just say other chunks of that. So the question of interest that we are trying to answer is chunk xj important? Does one of these matter in the linear models? And we're going to try and make this really, really general And so instead of talking about specific chunks, we're just going to denote this little curly, this little fancy A. And this fancy A just says, these are indices for a chunk of predictors we're interested in testing. So from here on out, we're only going to refer to this little curly A, and we're not going to talk about chunks, but essentially this framework allows you to do this testing for these chunks. That's the point of this. So we're going to need a tool to assist us in this. So returning to this full model, the tool we're going to use is this thing called a reduced model. And the idea is if I have this little subset A, what I'm going to do is I'm going to black those out, just take them out of the model, pretend they don't exist, and we're going to call this a reduced model. So the idea is we're just going to set beta j equal to 0 for all the j's in this little index set. And so you can imagine that it's just, yeah, just literally just take them out, just pull them out. And we're going to use this reduced model to answer a question for whether the variables in curly A were useful or not. So to frame it statistically, we're going to ask, does the full model improve on the reduced model? That's how we're going to answer whether this curly A variables are important or not. So this is testing a null hypothesis that beta A is equal to 0. So by beta equals 0, I just mean all the j's in A, those beta j's are equal to 0. So we're going to test this null hypothesis. So this is essentially the problem that Madeline and Garrett came to us with. So to summarize what they did, what did Madeline and Garrett do before talking to us? So the way they went about doing this is, what if we computed R squared for the full model and R squared for the reduced model, and then compared them? So we define this little delta R squared to say the difference in R squareds. And the idea is essentially when this delta R squared is really high, that means that the full model explained a lot more than the reduced did. So delta R squared basically says that the curly A variables were more important, is the way you can think about it. And Daniel and I thought this was an awesome idea. This is a great way to test whether the curly A variables are important, but we just want to augment this with a real actual hypothesis test. We want to get actual p-values with it. And so we do like delta R squared a lot, but we're actually going to use something just a little bit different, which we think will make this better for doing stats. So we're going to actually use this variable f. So if you've ever seen an f statistic in a linear model, that's this. And there's a few ways you can define the f statistic, but we like this way of defining it because it emphasizes its relationship with delta R squared. So you'll notice on the left, there is a delta R squared, but we have this little thing on the right, and this little thing on the right is just this extra factor for better statistical behavior. And so this little factor on the right will make this actually follow an F distribution under certain assumptions, which we'll maybe talk about later. So to talk about all the little things as part of this factor, that's just the number of variables in this set A that we're testing. And then once again, capital T is the number of time points, and then P is the total number of covariates in our full matrix X. So if we're going to go to classical statistics theory, like what you'd find in a textbook, there's a way to do this test. So the textbook way to do this hypothesis test that beta A equals 0 is under an assumption that your error terms, remember these from the first slide, if these error terms are independent and they're from a mean zero normal distribution with common variance, then assuming that the null hypothesis is true, this F value that we defined earlier follows an F distribution with these particular degrees of freedom, which is the number of variables A and then T minus P. Which means that with this approach, we can get P values under H naught. So we actually have a way to put quantified uncertainty on the importance of that A set. And if you're an astute viewer, you'd notice right away maybe one of the big problems with this, if we're going to try and apply it to neuroscience data, yeah, independence, that's super not true in this. So I drew this little plot of DF over F recordings, and these are definitely not independent. If I know the DF over F at a particular time, that tells me quite a bit about what it is in a little neighborhood of that. So we definitely do not have independence here. And so this unfortunately does destroy this whole F distribution theory. So our contributions are finding ways to remedy this with this particular type of data. So that's what we'll be talking about in this. So let's talk about what is kind of going wrong or to illustrate like why this is bad. So to do this, I did a little data simulation on my own. and I generated this data. And essentially I had y follow this sort of sine looking function thing with a little bit of noise added onto it. And then as x, I just had this slow drifting upward thing. And the key thing here is x was not used whatsoever to inform y. Like I did not generate y using x at all. So essentially there is no relationship between these two. And certainly not given that y is like, you know, this weird sine function, x is just this upward thing. But if you fit a linear model with this, you get an F statistic of 8.003. And with these particular degrees of freedoms, that gives you a p-value, which is super, super significant. So when you have the super dependent data, you get super, super artificially deflated p-values. And so you basically cannot be using these F tests to in linear models where like this dependence is there. So yeah, big problem. We're going to try and figure out how to fix it. Okay, so here's our idea. So we want to test this null hypothesis. And so our idea was, well, we cannot use the theoretical F distribution, but maybe we can approximate what the null distribution of that F value that we defined before under this null hypothesis. So our idea was, let's come up with a smart way of coming up with null neural activity, putting null in quotes because maybe that's not a super precise thing, but the point is that if we can generate null neural activity, we can kind of get a sense for what the distribution of that F little equation I wrote it before is under the null hypothesis. So as a little sketch of the algorithm we're going to be using, we're going to create capital B instances of said null neural activity. So for B equals 1 to capital B, we're going to compute this Y star B, which is just the B instance of null neural activity. Then we're going to calculate this F value that we defined before with this null neural activity. And then to get a P value, we're just going to count up the number of times in which this null F was bigger than our observed F, and then essentially divide it by the number of randomizations. And you'll notice there's this little +1 in there, and that's just to make sure we don't get p-values which are exactly 0 with this randomization thing, which help with certain simulations. But essentially the point behind it is we're counting up the number of times in which this null F is bigger than our observed F and dividing it by the total number of null things we got. So we're going to operate under this algorithm.
Question, Ethan?
Yeah.
In this version where you're computing this sort of empirical null distribution, is it critical that you're using the F statistic still as opposed to just the R squared, or does that still matter?
Yeah, that's actually really good. Yeah, a really good point. I think you probably, yeah, I'm not sure actually. I think you probably get something that's really similar with the delta R squared one. I think we liked the F one just because it's like what's used in F distributions, and we know how F distributions behave when these assumptions are met. So that's probably why we picked that. Yeah, I think you probably could use delta R squared. Yeah, I'm not sure. That's a good question though.
Thanks.
Yeah. So if you want to do like a randomization test or a test like this you could definitely switch what statistics you're calculating. So if you didn't think that an F distribution is right for your particular application, like whatever project you're working on, if you had some other statistic that you did think was a good one, you could use that under this framework likely. So so in this project we thought the F was a good one, but I think you could change that up. So with this framework, we didn't say actually how we're getting the null neural activities, and that was a lot of what we've been messing around with in this project. So we essentially had 3 ideas to come up with null neural activity, and the order of these is going to show our trajectory working through this project. So the first one, like our very first initial idea is, well, what if we just take our neural activity and we just shuffle it around, push it around? So to kind of visualize what this looks like, we're just taking this neural activity on the left and we're just totally shuffling it and getting the thing on the right. And this was sort of an immediate dead end for us. This shuffled neuron like totally does not look realistic. There's no way you'd see this thing if you're actually, looking at a brain under this imaging stuff. And you'll end up getting like super tiny p-values, if there's no association between x and y. So basically point is this just total permutation thing just does not work. So we're gonna put a red X over it. So our next idea was this idea called wrapping. And as a visual aid for explaining wrapping, the idea is we're going to try and destroy the relationship between X and Y by taking our Y and sort of wrapping it around. So I drew these little reference lines to kind of help you visualize this. So imagine on the left side, look at that chunk from red to purple. Just imagine taking that and sliding it to the left until the red is at the very left side of the plot. And then taking everything you pushed like sort of over the edge and just move it to the end of the sequence. So you'll get the thing on the right after you wrap it. And the idea is that if you do this at a sort of random index along here, the point is that you're destroying this association between X and Y. So this is like a quote unquote null Y. But it is realistically observable because you did actually observe this data just with a different sort of one weird breakpoint in it. So we think there's a few good ways and a few bad ways to do wrapping. And so we'll talk about that a bit. So our first option, which is what we were doing for quite a long time, is so if we just consider an arbitrary neuron, in this case, we'll just call it neuron 34, to get the p-value for this thing, we'll essentially wrap neuron 34 10,000 times to get 10,000 null versions of that neuron. And we were doing this for a while, but we don't think it's the best way to do it. Like sort of con of this approach is that what it means to be not responsive to or not associated with X is not fully captured with this. So if you imagine like if you imagine this, for example, if I were wrapping, if this was neuron 34 and I were just wrapping neuron 34 10,000 times, every single wrapping realization would have this big peak in it. And so I'm not ever allowing myself to have a null Y, which wouldn't have a peak in it. And maybe that's a bit of a problem. And maybe we'd like to have neurons which don't have peaks in them. So essentially we can't capture what every single neural Y would look like. So that's why we think this is a bit of a con. And so another way to go about doing this, which we think is a bit better, is rather than wrapping the same neuron a bunch of times, we'll take a whole bunch of other neurons and then wrap those 100 times each. And this will give us more diversity in our null Ys. So yeah, this is a better approach. This will give sufficient variety in the null distribution of F statistics because we're getting sufficient variety in the null Ys. So yeah, we think if you're going to do this, we think option 2 is a lot better of an approach. So yeah, so permuting is not good, wrapping is good if you do it right. And then our third one, which is a very recent exploration we've done, is using intertrial intervals to get this null Y. So what I mean by that is the idea is we're going to use DF over F recorded between trials as a sort of like proxy for what a null Y would look like. So I do this little visual aid here. So imagine we have this experiment happening and something interesting is happening. So that's why there's a spike. And then there's these little blue periods in between it. And these blue periods, there's just, you know, the experiment's not running, nothing interesting is happening. The mouse is just running around doing whatever, you know, nothing crazy. But we're still recording DF over F during this time. And the idea is that this is a null Y that's not associated with the experiment. So the idea is that we take all of our blue data and just stitch it all together. And after stitching it all together, we can call that a null neuron. And I just want to say, like, as a disclaimer, this is just a toy example for visual aid. I just took a trial and like repeated it. So you don't have to necessarily have peaks in the trials and peaks not in the trials. So you could have maybe peaks in the intertrial intervals and not a peak during the experiment. But the point is that in the intertrial intervals, supposedly something less interesting is happening than what's happening during the experiment. And that's what we care about. So we think this is a really good approach to calculating our null Ys. So with all these things I've introduced, unfortunately, there are some computational problems that we have to think about. And this is a good audience for that. This is a computational seminar, so we'll talk computation. So going back to our algorithm, our little sketch of our algorithm, we're actually not gonna think much about how to generate null y's for just a second. We're just gonna go to this little high level. So going back to this, first off, if I care a lot about accuracy and I want of null y's, this algorithm is going to be really slow if this capital B number is large. And as a second thing, if we've got a whole bunch of neurons that we're looking at, we have to run this little gray box algorithm that many neuron times. So now we're looking at 20,000 capital B plus 1 computations, or that's 20,000 times B plus 1 times we have to calculate our F statistic, because you have to calculate capital B of them for the null ones, and you have to add another one for the observed F1. That's a lot of linear models to fit. That's horrible. If you run this, it's going to take forever. So some of our contributions include fixing this with a few clever math tricks to take it down. So our insight was that if your capital X matrix is the same for all neurons in your experiments, all of the hard work in these computations can just be done a handful of times, or twice in this case, rather than 20,000 times B plus 1 times. And so this, if you only have to do the hard work a few times, this saves you a lot of time, which will make this a lot more powerful. So you might be asking, like, well, what is the hard work in this? So the hard work that I'm talking about is just fitting lots of linear models, because the idea is that if you do this naively, you're fitting a linear model every time you want to calculate an F statistic. You'd use Python or R or MATLAB or whatever and fit your linear model and then extract the F statistic, but you're fitting a linear model every single time you want to do it. And you can fit one linear model really quickly. You've probably ran an R or Python, like fit linear model, and it runs instantly. But when you're doing this, millions of times, that's actually really, really, really slow. So fortunately, there's some math tricks to help us with that. So the insight is that our F statistic, which we're calculating, you can also write it using a special residual, or we can use it using residual sum of squares. So as a reminder what that is, you just take the sum of these square differences of your observed Y and your predicted Y using your model. And so you can do this for your reduced and full models. And essentially what you can do is you can write your F statistic this way as well. So before I wrote F statistic as delta R squared times a factor. And in this case, I'm writing it this way, but they're equivalent. Mathematically, they work out the same. So now the point is that if I can get residual sum of squares R and residual sum of squares F for reduced and full, then I can calculate F without actually fitting a linear model. So the trick that we have is if your data matrix X has this singular value decomposition, then as a fast trick, the residual sum of squares in the linear model is equal to this little formula. So the norm of Y squared minus U transpose matrix product of Y, and subtract that. And that actually gives you residual sum of squares. So the point of this is that if I pre-compute the singular value decomposition of X, I can just keep reusing this U, and the only thing I have to plug in each time I want to calculate an F is this Y. So I can just plug in a null Y, plug in a null Y, but I keep U sitting around, and that saves me like a lot of time. I think using this trick saves you like hundreds to thousands of times of computation time, which is super cool. Yeah, so applying this data to the motivating data sets, here's a sort of little one of the results we got. So on the y-axis, we have Madeline's delta R squared, which we did think was a really great way to evaluate that. Then on the bottom, or on the x-axis, we have our negative log 10 p-values using our method, using specifically the ITI method of generating a null y. And if you look at this plot, there's a very, very good correspondence between what delta R squared was before and our p-values. And so we like delta R squared, but the p-values have quantifiable levels of uncertainty on them. And that gives you a more justified level of thresholding if you want to say, these neurons are associated with this chunk of predictors, but now you can put a threshold on it in a way that's quantifiable and makes sense. So in conclusion for this work, we showed a way to get p-values when your hypothesis tests look like H0 beta A equals 0, where A is just, remember, is just a subset of your indices for your predictors. And we did this for linear models when some of these classical assumptions like independence and IID from a normal distribution are not met. So our two contributions are we can get p-values, but not only can we get them, but we can do this really, really quickly with our computational trick. And of course, big thanks to Madeline and Superlab for the motivating data set. We thought it was a super interesting data set, and it was really fun to work on this problem. So that's what I have for my presentation. I'm very happy to answer any questions if anybody has them at this point.
Yeah, thanks, Ethan. That was a fantastic talk. Yeah, we definitely have time for questions.
I'll ask one. Yeah, awesome talk, very clear and very cool work. Is it an assumption of the ITI method that the autocorrelation of the signal that you get when you stitch together all the ITIs is the same as the autocorrelation of the within trial data? Like, I'm imagining a scenario, and this may be a rare scenario, so the real question is like, can you test, if that's the assumption, can the assumption be tested? I'm imagining a scenario where, like, you know, if you're doing one of these experiments like I assume Madeline is doing, the mouse is like getting rewards as they go on in the session. Maybe they're getting less and less thirsty, so their response to the stimuli or to the rewards is getting smaller and smaller as the session goes on. You might have some long autocorrelation in those in the within trial data, but that might not be reflected in any autocorrelation of the ITI stitched data.
Yeah, that's a really good point. I think that the assumption that is kind of being made with the ITI method is that sort of this distribution of y over this is the same in an ITI as it is in an experiment if essentially the mouse was not responding to certain like factors. And so I think that includes as a result the autocorrelation structure if the distributions are the same. So I'd say yes, probably. I would imagine that if the autocorrelation structure is close enough, then it's probably not much of an issue. Yeah, I would describe this method of getting a nullized as more of like a clever trick that we think is a good idea, as opposed to something that's, you know, super proven. If that answers your question, but I would say probably it does assume that there's like a similar distribution. Yeah.
I have a question.
Yeah.
Had you guys thought of, like, what about instead of shuffling your Ys, you shuffle the columns in X if you're trying to shuffle something like trial identity? I mean, does that work with these methods you presented here? And is there a reason you guys didn't try that as well? And shuffling the Y?
So we actually did try that. Let's see, let me pull up my slide where I have my models written up. Hopefully you can see my introduction slide. To repeat the question, basically, like, why are we shuffling Ys rather than shuffling our X's or doing something with X rather than Y? And we did actually look into that. And I think recently we decided that probably shuffling X is a bit better, honestly, just because if we're shuffling our Ys, now we're destroying the association between Ys and all the variables that do matter in X. Whereas if we like only shuffled the, see if I go to like, so if this little a that I'm taking out, if I were to only shuffle the X's in there, then I'm only destroying the association between Y's and the X's in there. So when we were first thinking about this, we did actually try shuffling just those columns of X as well. And just empirically, we saw that the like p-values you got from the methods were like really, really, really closely aligned. So I think we went with just shuffling the Y's because you have to do less singular value decompositions. Because if I'm shuffling X, then essentially I have to do an SVD for every single time I shuffle X in some way. And maybe that's not too bad, because if you shuffle X in the same way for all of your different null X's that you're getting, then you'd only have to do like that many SVDs. Whereas with the Y, you only have to do 2. So maybe it's not that big of a deal. Like it would add on a lot of computational work, but I think maybe that's probably the better way to do it.
So sorry to interrupt you, but just to answer that like a little bit more directly, I think it is like slightly statistically better, as the person who asked the question pointed out, to shuffle the block like the thing that Ethan's calling the chunk rather than shuffling Y. Like statistically, that's a little bit better, but empirically it seemed to make no difference. And we just thought that like probably Madeline, if there's no empirical difference, would rather have her code run 20,000 times capital B or whatever, 20,000 times faster, if 20,000 is the number of singular value decompositions that you save. So basically we checked with Madeline, how do you feel about a 20,000 times speed up for no empirical difference? And she said she'd be happy with the 20,000 times speed up.
I'm very, very happy with how fast it runs. I'm running my whole model for like hour-long sessions, 500 neurons run in less than 5 minutes. It's really impressive.
Awesome. Yeah, that makes sense. Thanks.
Yeah, really good question. Also, I'm happy to hear that Madeline's happy with this.
Have you, that's another one. Have you thought about like how do regularization methods or yeah, come into this? Like,
Yeah,
have you thought about, yeah,
that would definitely get a
what
bit
to
more
do?
tricky because I think that a lot, well, at least with, well, okay, so for the null Y thing, that wouldn't matter at all. But for this computational speed up, this little, where did I put it? This thing, this RSS thing only works if we're like not putting a regularization on this linear model. So I think maybe like under certain regularization schemes, like maybe if you're doing some type of ridge thing, there is a way, some like analogous form of this using the SVD. I don't know of it off the top of my head or,
Yeah,
so
I think that if you're doing, sorry to interrupt, if you're doing
Yeah.
ridge regression, then a comparable trick would work. But if you're doing like a lasso or an elastic net or anything with an L1 penalty, then like it just wouldn't, this wouldn't go through. So you would have a lot slower computations. So we were really happy that for this particular project, Garrett and Madeline didn't seem like super interested in right now using regularization because it meant that we could do it fast. But I think for ridge, it would be fine, ridge regression.
Yeah. But like if you were okay with the slower computation time, then I think the same method, at least with this like algorithm I outlined, this would work just fine, even if you're doing some other model.
I have two questions. One is more so about like the pre-processing the data. So when you're stitching together data from different neurons and ITIs, sometimes there are like these discontinuities in terms of the values. Like maybe like in one ITI, it's like at a like low like activity, and then the next one there's like a, it's like in the middle of a activation that at the start of that other ITI. So I'm just wondering like how you accounted for that.
Yeah, I think that was something that we kind of just had to be okay with. So if I go to, yeah, here, so you're saying like, let's say the first blue one's low and then the next blue block, it's a lot higher or something. So then once you stitch it, then you get this big jump between the two stitched blues. Yeah, I think that's something that's maybe a bit imperfect about this, because I think if you did have a really high blue, then you would have this kind of jump in between it. So I think you just have to be okay with that. I will say
wouldn't you just shift it down to make them continuous? Like this is just kind of like fake data that you're stitching together. So you could just.
So yeah, that's true too. You could also just, if you wanted to, if you were really worried about that, then yeah, as Danny said, you probably could just shift it down. Yeah, I personally did not do that, but you probably could do that.
I guess one other thing to, sorry to not be Charles, but just one other thing to say about this, like this ITI thing, like in Madeline's data, like she had these intertrial intervals. And so we were like, let's just stitch them together and see what happens. But like, if you have an experiment where you like started imaging and you imaged for like 5 minutes before the trial began, you could use that instead of the intertrial interval. Like we're not specifically attached to the intertrial interval. The spirit is we just wanted something that's like nothing interesting happening with respect to the covariates. And we got that from Madeline's data by stitching together ITIs, but for a different experiment, you do it differently, presumably.
Yeah.
Based on the design.
Yeah, really good point.
Yeah. I mean, I guess it's also the question of if those discontinuities actually make much of a difference, which I don't really have expertise to say about. And I think just kind of piggybacking what you're like responding to me about is that I kind of like the neuron stitching, because if there is a like activation at the beginning of the session, then you can kind of just and I guess this works if you have that like 5-minute or like a small period of time at the beginning of the session where it's not animal's not doing anything. You can just like cut it off to a point where there's not an activation or something. And the second question is actually, sorry if it's addressing something that you didn't really present here, but it's been something on my mind just when we first started thinking about this model, is that the number of features in your in the model, as I remember, there's a lot more features than the samples, which is like the amount of time points. Because I think that to account for like delays in the calcium activity relative to the behavior, it's like multiple kind of shifts for each of the columns for each of the features. So I'm wondering if like overfitting is something to be concerned about. I just don't know.
Let me make sure I'm understanding correctly. So are you saying like what do you do if like capital T is less than P?
Yeah, and I guess is it even something to be concerned about?
So in this particular data set, that was not something we dealt with just because we had like 17 something thousand time points and then like 200 covariates. That wasn't something we really thought about.
Okay.
So me answering this is like totally off the top of my head. I haven't even thought about this before. I mean, obviously if like capital T is less than P, then you're not going to get like a unique linear model or not a unique beta vector that, for your linear model. So you probably would have to, I know there's things like minimum norm solutions of beta for linear models. And I know there's also like, if you do like lasso, now it becomes unique. So you probably have to think about that if you're going to try and answer that. Yeah, I don't know.
So I think I would want to, instead of fitting a least squares linear model, which is what Ethan did, I would use ridge regression. And then I believe I wouldn't use the lasso for various reasons. I wouldn't use the lasso with elastic net. I'd use ridge regression. And then I believe that the computational trick, I think it would go through, although to be honest, I haven't checked carefully, but I think it would. The computational trick that allows you to just compute one SVD instead of having to fit a million linear models. I think that would go through. The exact equation for it would change, but I think there would be a trick like that.
And in terms of whether like your linear model would like somehow overfit, I actually just don't know how to answer that. I don't really know what the effect of it would be. So like if T was really close to P, say, I'm not sure.
Cool. Thanks.
Yeah.
Okay. If there's no more questions, then let's thank Ethan one more time. Yeah, I really enjoyed it. You, as I think someone else said, is very clear and very appropriate for the audience.
Thank you. Appreciate it. Thanks for having me.
All right. See y'all next time.